Hi, everyone. I'm Kathleen McIlroy. I'm the Associate Director of the School of Journalism here. Moody College is so ecstatic about having this talk. Richard Harris has written a book on a topic that's very much in the news. If you read the New York Times Magazine, I think a couple of weeks ago, they did a whole cover story on the reproducibility project. And it's the idea of, can science be reproduced? Or is this one study just a phenomenon in and of itself? The very first line of the book he just produced, Rigor Mortis, and How Sloppy Science Creates Worthless Cures, Crushes Hope, and Wastes Billions. He writes, when you read about advances in medicine, it often seems like long-awaited breakthroughs are just around the corner for cancer, Alzheimer's, stroke, osteoarthritis, and countless less common diseases. But it turns out we live in a world with an awful lot of corners. So I like this idea that we've been waiting for these breakthroughs. What happens when it seems like this isn't really happening at the pace we think it should happen? So I am very, very pleased and to have Richard Harris here. And I want to thank the American Association for the Advancement of Science and the Kavli Foundation for making this AAAS Kavli Science Journalism Award lecture possible. Now, this is important also because AAAS is having its big convention here in Austin in February. So science and science journalism is all in the news, and it's going to be around us. Also, really want to thank the Kavli Science Journalism Award lecture series. It allows us to have people, distinguished journalists like Richard Harris here. These awards were established in 1945, and they've gone to some of the most notable writers in the United States, including Rachel Carson and Isaac Asimov. If you're an undergraduate, you may need to Google those names. But I suggest you do, okay? Rachel Carson and Isaac Asimov. Actually worked with one of his relatives. The Kavli Foundation endowed the awards in 2009 and doubled its commitment in 2015, opening the contest to journalists around the world. And I think that's particularly important because we now know what happens in Malaysia, what happens anywhere is affecting us. We need to be less United States centric, even though some of our officials may feel otherwise. Richard Harris, tonight's speaker, has won the AAAS Kavli Award 3 times for his work at NPR, including a series in 2010 about the blown out BP oil well in the Gulf of Mexico, spewed far more oil than the officials had actually estimated. He's traveled to 7 continents for NPR, including this cool photo where you're shown by the icebreaker. I don't know if that's in the Antarctic. He's been Timbuktu, South Pole, Galapagos Islands, Beijing, doing SARS, You're a Brave Man, the center of Greenland, Amazon rainforest, all kinds of good stuff. In 2014, his focus shifted from an emphasis on climate change and the environment to biomedical research. And as I said, his recent book is Rigor Mortis, How Sloppy Science Creates Worthless Cures, Crushes Hope, and Wastes Billions. There's some flyers around. Don't just sign them, have me sign them, but actually pay attention to what the flyers say. So his talk this evening is titled, Bad Science, Good Science, Covering Medical Research. Welcome, Richard.
I will give you we have a clock and everything, so that's good. I will give you a brief tour of bad science, good science, and sort of how my thinking has evolved on covering science since I was asked to come back and cover this topic a couple of years ago and ended up writing a book on the topic. Now, I'm going to start with a cartoon that's 20 years old, but is still meaningful today. This is the random medical news. You spin this wheel and you say, coffee, it can cause depression in twins. And so according to a report released today, he says, and then they spin the dials. But who hasn't had this phenomenon, felt this phenomenon where basically every day it's some other disease, some other thing, and it's either good for you or bad for you and whatnot. And why does this happen? And that's a good question. And what part of the reason it happens is because science keeps changing and evolving and improving. If you've heard of CRISPR, that's a new technology that allows incredibly precise gene editing. And it replaces some technologies that did that pretty well, but not so well. And this paper that was published earlier this year reports that scientists are realizing when they did the same experiment with this more precise and effective tool, they got different results than when they'd done it before. So it's like, okay, out with the old and in with the new. But it also means a lot of the research that they'd done previously wasn't giving them the results they actually thought they had. So that's one thing. Technology always changes. Science changes. Science is a process, right? And we improve things and we get deeper understanding. Another reason this happens is science is full of bias. It's a human endeavor the way everything else is. Our lives are full of bias. We all have confirmation bias. We seek out information that supports our point of view, and we tend to downplay things that don't. And so this is a paper that was published a couple years ago, and John Ioannidis and his colleague decided to look through the literature and see how many different kinds of biases are mentioned in the biomedical literature, and they came up with 235 different kinds of bias that scientists report, which means, and any of these things can mess up the way you interpret your experiment. If you haven't, for example, been careful to blind your experiment so you don't know whether you're looking at the experimental set of animals or the control set of experiments. You can fool yourself into thinking you have one result when in fact you don't, when you have a different result. And confirmation bias, all sorts of other biases crop up in the literature. And actually science, Richard Feynman, who's a famous physicist, gave a commencement talk at his university where he was teaching Caltech some years ago, and said, basically, what science is really all about is a set of principles to prevent you from fooling yourself. And he said, and you are the easiest person to fool. So reducing bias is a fundamental part of science. So here's another problem. This is another paper, same author actually. This is an essay he wrote some years ago. Why most published research findings are false. It's like, how can you say that? And the answer is, well, some people said, well, they didn't entirely buy the argument. But his argument is that so many experiments are small that the effects that they see, if you repeat the experiment, actually wouldn't show up again. But scientists don't often repeat those experiments. And they actually don't understand the statistics that go into the experiment. So in science, there's this whole cult of the p-value. And if a p-value is less than 0.05, which is 5%, then scientists say, okay, it's a real result. It means And some people misinterpret that to mean there's only a 5% chance that it's wrong and a 95% chance that it's real. That's actually a very poor interpretation of what p-value is. But a lot of people do that. A lot of people run these kinds of studies. And so as a result, he argues that particularly small studies, that the vast majority of small studies, if you repeated them again, you would find out actually they don't hold up. And so this is another argument that there's some misunderstanding about how to interpret results once you have them. So this paper really caught my attention. This was published in 2012, and when I started my new beat at NPR on biomedical research, I came across this paper. This speaks to how big a problem this is. And there are more than 1 million papers published every year in the biomedical literature. So there's no way to check and see how reliable that entire literature is. But Glenn Begley, who was the head of cancer research at a drug company called Amgen, decided he wanted to look at papers he really cared about, papers that were published in the literature that had come from academic labs. And he wanted to know whether they were correct, because if they were correct, they were important potential leads for new cancer drugs. And so he went and he dug up 53 of these papers that had crossed his transom over the years. He'd been at Amgen for 10 years when he did this work. And he said, how many of them can we reproduce in our labs? If we try to do the same experiment, do we get the same result? And he tried, and he did not have very much luck at all. So he took some of these experiments back to the original researchers and said, I'm having trouble reproducing these experiments. Would you do it for me? Can we come and watch and see how you do the experiment? And even the original researchers very often couldn't reproduce their own results. And in the end, of these 53 studies he looked at, he could only get 6 to reproduce. That's what, 11%? And that's a pretty low success rate. There was another paper that was published around the same time from scientists at the drug company Bayer, and they found that they could reproduce 25% of the papers that they were really interested in. So this led people to be concerned about, wow, how reliable is the biomedical literature? How much can in fact be reproduced? And I must say that this paper itself is not reproducible because he didn't say what the 53 papers were. So nobody can go and check his work and see if he was right. And it's possible that some of his failures were his fault, not the fault of the original paper. So that's another important lesson. It's not easy to tell. Even if you try and fail, you don't know necessarily which one is right and which is wrong until time elapses. So this is a problem that is not just in biomedicine. This is a paper that came out in Science a little while ago. And this paper looked at reproducibility in the psychological sciences. And Kathleen mentioned the article in the New York Times Magazine about a social scientist named Amy Cuddy, who had done some work. She has the second most popular TED Talk on the planet. And she basically makes this argument that if you do these power poses, I don't know what they are, but basically it makes you not only more focused and energetic, but it actually changes your hormone balances. So actually it does make you more powerful if you do these power poses. And she ran into trouble. It turns out that her findings were not reproducible. And in fact, when somebody did a bigger and more careful study, they didn't see those results at all. But but she was actually following the general rules of the psychological literature of how psychology experiments are run. And it turns out they're run to a fairly low standard, because this was a paper that tried to reproduce 100 studies that they sort of picked because they were interested in the 100 studies to see whether they could reproduce them. This was another group of people and talked to the original researchers and said, if we try it this way, would you expect, it's not an identical experiment, but would you expect that we should get the same result? And of those, only about a third of them were able to be reproduced. And some of them, about another third of them, were actually came to the opposite conclusion. And a lot of them were just kind of muddled. It was like, well, it might be something, but it's not a significant result. And so this paper made a lot of headlines as well about raising questions about the reproducibility of the psychological literature. This is another example of just sort of a tap on the shoulder. How many of you have heard these stories about a new gene for schizophrenia has been discovered, right? We've heard many of these over the year. In fact, these guys went and looked at 25 genes that have been published, and it's like, oh, here's a gene that's associated with schizophrenia. And they went back and took another look, and they said, actually, on further reflection, none of these 25 genes has anything to do with schizophrenia. So it's a problem. Now, how often does this creep into journalism? And this is a question that actually some academics have taken on. Estelle Dumas-Mallet and her colleagues at the University of Bordeaux published this analysis earlier this year. And what they did was they started out looking at 100 English or 200 English language newspapers from around the world and dug through them to see what scientific breakthroughs, particularly in medicine, were announced in these newspapers. Things like an association between a particular pesticide and Parkinson's disease, for example. Or there was one paper they looked at that reported a new mechanism to explain ADHD. And they said, well, let's find out how many of those actually stood the test of time, because these were all, a lot of them were initial studies, sort of suggestive stuff, the kinds of stuff that grabs headlines. But what happens as science continues to chew away at those topics? What happens? And they found 156 of them that had, where there had been enough information accumulated about the topic over the years that scientists could go back and do what's called a meta-analysis. They could gather all of the published data together and do one big analysis and sort of imitate what it would look like if they'd done one big study instead of a bunch of little ones. And they found that when they looked at these 156 studies, the papers, there were about half of them showed that the findings stood up over the test of time, and only about 34% of the most exciting ones actually stood up. So it's like if you have a particularly exciting finding, you have like maybe a 1 in 3 chance that it's actually going to stand up upon review by further science. And do we as journalists report oh, by the way, that what we said about Parkinson's disease and pesticides, that turned out not to be true? Now, we actually do a pretty poor job of following up on that, partly because the initial studies are published in big journals where they get a lot of attention and so on, and then the follow-up studies tend to get published in smaller journals that journalists don't have the wherewithal to dig through and find out. So we may not even realize that a lot of the things we've reported on turn out not to be true down the line. And it's also kind of weird, if you think about it, to do a story that says, hey, remember that story that we published 6 years ago? Probably not. Well, it turns out it wasn't true. Thanks very much, you know. So there's some other reasons why we don't report those. But it's important to remember that those are often dodgy studies. So this all led up to last year, Nature decided to say, you know, ask scientists themselves. So is there a reproducibility crisis in science? And this is of all sorts of scientists they asked. 52% yes, there's a significant crisis. Another 38% said yes, there's a slight crisis. I'm not sure what a slight crisis is, but it was a lot of people like that answer. And 10% said, I'm washing the dishes, I don't have time to answer your question, or I have no idea, or whatever. But the point is that this is an idea that has really taken hold, particularly in the biomedical community, but elsewhere, that there is actually a crisis of reproducibility. I argue there's actually not really a crisis. There's not something new that's happened that's just cropped up, but I think what we're seeing is a realization that science is a lot less reliable if you're looking at the tiny granular level of study by study by study. It's a lot less reliable than we have understood, and we need to be much more cautious in interpreting any single result. And the world of science also needs to think about ways to improve the reliability of these individual stories or studies. And I will talk about that a little bit before I get to what journalists can do as well. So not only does the general pile of scientists agree this is a problem, but Francis Collins, who is the head of the National Institutes of Health, much to his credit, when he saw these issues emerging, instead of just sweeping them under the rug, he said, yeah, we have to deal with these issues. This is, you know, I'm the steward of $30 billion a year of taxpayers' money. And if we have problems, we have to figure out what they are and how to fix them. And this is a quote from a paper he wrote with his chief deputy, Larry Tabak. And he explains, in the long run, science is self-correcting. That's the beauty of science, right? The scientific process is trial and error. You find things. Eventually, the truth sort of filters out. But he says, but in the shorter term, the checks and balances that once ensured scientific fidelity have been hobbled. Pretty strong words from the head of the National Institutes of Health, right? So at any rate, but after sort of getting that general gestalt, I decided, okay, I'm going to write a book about it. So I took a year off, and thus was born Rigor Mortis. Actually, my preferred title, my working title for the book was Science Friction, because the idea being that these are problems that are slowing down the progress of science. Science isn't dead, or rigor in science isn't dead, as Mortis might suggest, but science is suffering from needless friction, and my book explores ways to deal with that friction, to minimize it so science can move ahead faster. So what are the causes of this? Well, bad ingredients in biomedical research are quite common. Designs that are susceptible to bias crop up more than you would care to believe. Statistical errors are another really big problem. And the funding pressures and sort of the underlying culture of science, because the NIH budget doubled between 1998 and 2003, which created like a huge outpouring of new science buildings and a whole bunch of hiring in the sciences. And then since that time, over the following decade, in real spending power, Congress said, okay, we've done enough, NIH, well, let's turn our attention to something else. And in spending power, the funding decreased by about 20%. So all of a sudden you build up this whole new enterprise and then you start to starve it. So that creates a bunch of incentives that drive scientists to do things that they have to do to keep their careers going, but it's not in the best interest of science. So this is a photograph of Henrietta Lacks, who in 1951 was diagnosed with cervical cancer. She was at the Johns Hopkins Hospital, and she, the doctors there were able for the very first time to isolate some cervical cancer cells from her and keep them alive and perpetuating indefinitely in a petri dish. And she's, I don't know if you saw the Oprah Winfrey show about her, or if you read Rebecca Skloot's wonderful book about the immortal life of Henrietta Lacks, which was the title of her book. But her cells became this incredibly useful laboratory tool because they keep growing and growing and growing, and anyone who wants to study cervical cancer in particular, or sometimes just cancer in general, can get her cells and use them. And the problem is that her cells have taken over the world. They've turned out to be sort of the kudzu of the biomedical world because they grow so well, so much better than any other cell, that they're an easy contaminant, and they're very frequently moved from one set of petri dishes or whatever to another, and before you know it, scientists thinking they're working on one kind of cancer has actually had their cancer cells overtaken by these HeLa cells, and they're actually just studying this immortal line of HeLa cells. And it's a huge problem. This paper, published just a couple weeks ago, took a look at how big a problem it is, how cell line misidentification contaminates the scientific literature. And these guys went back and they said how many different cell lines there are that are called one thing are actually HeLa cells. And they identified there are well over 100 different cell lines that are called one thing but are actually HeLa. And what they discovered was that in the scientific literature, just for not a complete search, but their one swipe across the scientific literature, they found 32,000 papers that were published that were using HeLa cells where the scientists themselves thought they were studying liver or some other kind of cancer. And then they said, how many people cited these 32,000 papers? And they found 500,000 papers in the scientific literature that did that. And that's just one kind of bad cell line. This is the International Cell Line Authentication Committee, and they've cataloged more than 400 misidentified cell lines. WISH is actually HeLa, Chang liver is actually HeLa, intestine 407 is HeLa, but a lot of these are completely different things. MDA-MB435 was isolated at MD Anderson, and it's been used for many years as a breast cancer cell line, but it turns out that very early on it was contaminated with a melanoma, and people today continue to publish these and call them breast cancer cells, even though it's pretty clear from research that it's actually a melanoma cell. And since that was discovered in around the year 2000, there have been 900 publications of this cell line calling it breast cancer when it's pretty clear it's not. So this is a problem that needs to be shaken. The biomedical researchers need to be much more attuned to this problem, and they're starting to become aware of that. And I'll have more to say about that in a minute. This is a man named Tom Murphy I met when I was doing some of my early reporting, as I was sort of exploring the new world of biomedical research in 2016, new to me. He had been diagnosed with ALS, Lou Gehrig's disease, which is a terrible muscle wasting disease, and eventually, in just a couple of years, 4 years usually, after diagnosis, you die from this disease, and there's no treatment for it, no effective treatment. There's one approved drug that has a marginal benefit. But he was told by his doctor, you could get on a drug trial. You could try something. There's a couple of options for you, and you can see if an experimental drug is going to help you. And he decided to sign up for a drug trial studying a drug called DEX. He got on the drug trial. He actually said it made him feel better, but overall, when they looked at the 1,000 people who were on this drug trial, it turns out that it really wasn't helping them, and so they said, "Oops, this is not working. Sorry." And it was yet another long list of failures for ALS. And this is a paper that you don't have to look at all the details, but the blue lines represent scientists who had been trying to develop these drugs. These were all ALS drugs. And the blue lines represented their initial reason why they thought this could be a good drug to study in human beings, because these blue lines show how much survival increased in mice when they were given these various drugs. And you can see some of them seem to do a pretty good job in improving survival in mice. This was his drug, DEX here, and it had some marginal, some improvement in the mice, which encouraged them to try it in humans. But this group called ALS TDI decided to take a close look at this data and see why none of these worked if all this mouse studies seemed to suggest that it would. And the first thing they found was that many of these mouse studies were very poorly done. They may have had 6 or 10 mice only in these studies, a very small number. People weren't careful to do some really basic things, like if you're studying an experimental drug in mice, you should make sure that when you're randomizing your animals into one group or another, that you break up the litter mates so you don't have all the pups from one litter in one group and all the pups from another litter in another group, because then the difference might just be between litters as opposed to in the drug. And so these guys said, let's do all these studies again and do them correctly with large numbers of mice and following all of these necessary things to avoid inadvertent bias in these studies. And when they did them again, that's what the black bars show. Basically, none of them really worked. And the catch was each one of those experiments cost several $100,000. And if you're and these guys had the money to do it, but if you're an academic researcher interested in ALS, it's really hard to get that kind of money to do an experiment. So you would just try. You'd just say, well, I can do it with 10 animals, and I'll get these sort of results and cross my fingers, call it a pilot study, and hope that the pilot study actually represents what's really going on. But that's again part of the problem that we're facing in biomedicine in this country, that there's so many people chasing a shrinking pool of dollars. It makes it difficult to do the science the right way, that way it should be. So that's an example of experimental design. Let me talk about some of the statistical problems that crop up frequently. And this is an idea that's called harking, hypothesizing after the results are known. And this doesn't seem like that big a deal when you think about it. You do an experiment. You're trying to see if this drug is going to affect heart disease or whatever, and it doesn't really seem to affect heart disease. But then you look over here and say, oh, look, all these people, there's a reduction in cancer in one group of patients, of one particular kind of cancer. Wow, maybe and publish a paper saying this drug reduces the risk of cancer. It sounds kind of innocuous, but it's actually a very bad idea. And to explain why, I have found this cartoon just a couple weeks ago in my newspaper. Oh, I see it's copyrighted 2012, but it was just ran in my newspaper a couple weeks ago. But here's the hypothesis, the scientist firing off her hypothesis and off to see what her results are. And she, that's basically what harking is. After you know what your results are, then you say, that was my target all along, right? So that's the problem. And this happens very frequently in science. It's still valuable to make those observations, but then you shouldn't say, this data support my hypothesis. You say, this data give me an idea for a new hypothesis that I now have to go out and test and do it correctly. Otherwise, you could just be playing these kinds of games. Scientists themselves are finding that in this hyper-competitive environment, they are more and more inclined to talk up their research, because their tenure may depend upon it, new funding may depend upon it. This is a study that looked at language in scientific papers between 1974 and 2014, just to look at how much hyperbole the scientists themselves were adding to their papers as a way of measuring how much of science itself is sort of being driven by this hype, this need to prove that I've got the latest, greatest, whatever thing in order to get my next grant and so on, and the temptation to sort of exaggerate results as opposed to saying, this is an interesting result, but I don't want to go too far. No, nowadays it's like the words robust, novel, innovative, and unprecedented increased in relative frequency up to 15,000%, which is itself a little bit of hyperbole, right? 15,000%. But the point is that the scientists themselves are trapped in this system, and they themselves are hyping their own work, and that's something that we as journalists have to be very aware of as we talk to them as well. In terms of the sort of the cultural issues that all this causes, there's lots to be said, but I will just touch on a comment from Henry Bourne, who's retired at UC San Francisco. And in his view, the real problem in this hyper-competitive environment is scientists are having trouble balancing ambition and delight. That you need ambition, you need to keep food on the table, you need to keep your lab going. Many scientists feel like if I can't get a grant, I may have to lay off my graduate students. They're feeling a huge amount of pressure in this very competitive environment. And universities used to be heavily supported by states. They are much less these days. State money is dwindling, and so scientists are more and more dependent, and research is more and more dependent on getting federal grants, and that creates this incredible pressure on scientists. And what he says is scientists should be driven by delight of understanding how the world works and driven by their own curiosity, but instead the balance is off, and they are driven by too much ambition, not just because they are personally, out for riches and fortune, but just you have to be ambitious just to keep your lab alive, and that's a really difficult situation for scientists to be in. So what are the solutions? Well, one thing is to validate your ingredients and the, oops, I thought I had another slide in there. Actually I do. And this is Carolyn Compton, who is a pathologist at the University of Arizona. She was up at the National Cancer Institute for many years. And what she argues is that a lot of samples being gathered in pathology, for pathology use in surgeries and so on, end up being put into science as a way of studying basic biology of disease. So somebody who has colon cancer operation, a little piece of colon cancer is removed from their colon, that tissue may go immediately into preservative or so on, or it may sit around for hours or even days before it actually gets treated correctly. And that variability is a huge problem for scientists who then look at that tissue and say, this is what colon cancer looks like. It's like, well, this is what this particular sample collected this way looked like. But she's been trying to get pathologists and surgeons and so on to collect these tissues with great care and uniformity so that they actually are comparable. Another thing that the NIH is doing is it's now insisting that scientists validate their cell lines. So those kinds of HeLa problems we hope will eventually be brought under control. Scientists say, now when they have their grants are told that they're expected to do that, they have to find the money to send out their materials to get them tested. The checks and balances are that whole system is just getting up to speed, so there's probably still a lot of problems in the system, but ultimately scientists are at least now on notice being told you have to validate your cell lines to make sure that you're using the cells you think you're using. Transparency is another very important way of dealing with this, and one feeling is that if you have scientists know that they have to put their data out for other scientists to look at, and the computer code that they use to analyze their data, that there will be a checks and balances on that, and scientists will realize, I better be sure I'm right. If I've got mistakes in my data, I want to find them, and I don't want to put them out for other people to find. So transparency as an idea of not of preventing errors necessarily, but of speeding up this process of the self-correcting nature of science. If the sooner those problems are discovered, the better off you are, because then you don't have people running down a bunch of blind alleys. So this is just one small example. There's a group in Charlottesville, Virginia, that's got this open science framework, and they are encouraging scientists to dump all of their data into it, and it's sort of a big data sharing system that they're encouraging scientists to use. I'm not sure it'll succeed because it seems like many scientists think it's more work to do it that way than the old-fashioned way, but they're pushing. They're trying to find ways to get people to be more transparent with their data and their processing, the processes that they use to analyze it. Better training is also important. In biomedicine, very often scientists graduate students and postdocs learn from the people who are running their labs, and their mentors may or may not have a lot of time to do that. In fact, many mentors spend most of their time writing grant proposals these days because money is so short, and you have to write 7 grant proposals to get 1 grant typically. So and the training in this preclinical research tends to be fairly thin, and so the NIH in 2014 sent out a proposal saying to all of the requests for information to universities across the country, saying who's got the best training classes, so we can share those ideas, that information, in this kind of training people how to do these kinds of preclinical biomedical studies, and they got essentially no answers. No one had a really strong course in training people how to do that. So the NIH is now funding some development of some of those courses. I encountered this wonderful guy named Arturo Casadevall, who's at Johns Hopkins, and he's decided that he wants to spend his focus getting scientists to think much more carefully about these issues, thinking about how to design an experiment. He says so often if scientists get an experiment, they can't understand the results. The first thing they'll do is run a second experiment. He says people should stop and think and be, a PhD has the word philosophy in it, right? He says he wants to bring the philosophy back into this world so that you can bring more knowledge and information out of an experiment and not just say, oh, that one didn't work, I'm going to try something else, and really try to think about where you're going with your experiments. And finally, changing the culture of science is a big challenge. You have to somehow or other figure out how to break this cycle of the hyper-competitive environment driving scientists to publish flashy papers that may not be right, and and they need to do that in order to get their tenure and their grants and so on. So that's a big problem to solve, but ultimately culture can change, and it does change, but it changes slowly. And there are also people thinking about how to make that happen. I want to say just a couple words. This is actually, this was at the National Academy of Sciences earlier this year, a colloquium on reproducibility of research issues and proposed remedies. And a lot of people were talking about some of these things at that. And I just wanted to focus on one guy named Trevor Butterworth, who runs an outfit called Sense About Science. And he was arguing that we really should make sure that journalists understand much better about where all the pitfalls are in science, so that they can cover science much more carefully and they can get it right. And I stood up after his talk and I said, I mean, I can correctly report what some scientist has put in the literature, but I'd like to remind you that a lot of what's in the literature isn't right. So it's not just on me as the reporter, it's on the peer review and all the entire scientific process to be more careful and more thoughtful about how they put science out there. I mean, we also have a responsibility to portray it correctly, and that's where I would like to end. What we can do, the first thing is don't assume that what you're reading is right just because it's published, even if it's published in a peer-reviewed journal, which is of course what first question we're always asked is, was it peer-reviewed? Yes. Even if it's peer-reviewed, it doesn't mean it's correct. Second thing you should do is ask questions. Are there other studies like this? Because science doesn't usually proceed from just single outstanding weird experiments, but it's accumulation of data. So are there a bunch of other studies? Where does this fit in? Is this the first one of 10 that found this result? Or does this support the results of 9 other studies like this? Another question is, are the statistical findings barely significant? Is it sort of really close to that magic p-value of 0.05? And if it is, there's actually a much higher probability that the paper is wrong than the scientists themselves even realize. So if you see those kinds of p-values in a paper, and you have to learn how to read a paper to find those p-values, but it's something even something you can pick up. That is that's a red flag for me. If the p-values are barely at that mark, it's like this doesn't seem like a very robust finding. And then are there other findings in the literature that contradict the result? If so, that could be interesting to explain why. Every study is different and looks at things in a little bit different way. Why are there conflicting results? And I think it's also really important on us to express uncertainty when we're reporting on things and not just say, here's the newest finding, coffee causes wrinkles, or coffee doesn't cause wrinkles, whatever. We need to do the best we can to convey to our audiences that science is a gradual process, that each piece is just one more piece of the puzzle, right? More than 1 million papers published every single year. Think of how much science is going on all the time and what tiny increments it's coming out in, and we shouldn't oversell any one particular finding. There are occasions where one finding is spectacular. There are these eureka discoveries that we report, and they are worth reporting, but those are fairly few and far between, and we should recognize that that's not the everyday kind of science reporting, but that's a pretty unusual occurrence when you can say, wow, we now know that the universe is expanding at an accelerating rate or something like that. These things don't happen very much. If the effect is small, you should say so. And this is a tricky business in biomedical research, because sometimes scientists kind of bury this information. They say, oh, this is a 40% increase or whatever, and 40%, if you actually dig down to the original numbers, 40% may mean little or nothing, and particularly if it's a small study to begin with. It could be the difference of 2 or 3 people, and if you just had results different in a small number of people, you could have had a completely different conclusion from a paper. So sometimes you have to look in the supplementary parts of the paper, unfortunately, to find out how many people they studied and how big the effects are. But it's worth your while to dig down and find those things, because percentages are a pretty poor way. If it's a 1 in a million chance of something happening and that increases by 50%, it's like a 2 in a million chance of something happening. It's still not something you have to really lie awake worrying about, right? So, or if that affects doubles, let me get my statistics right. Check your math. Never do math in public, actually. That's another rule that I just violated. The point is, look at the raw numbers. That will really help you tell the story and understand what's really going on. Because scientists might be, using some of those robust and extraordinary words in their abstracts, and their findings may not actually support that particularly well. And finally, follow up. Look back from time to time on big stories and see if they're panning out. In fact, there was a paper that came out just a couple of weeks ago looking at engineering human embryos using this technology called CRISPR. And there's a lot of news coverage about it and a lot of excitement about it. I've started to see bubbling around in sort of the area where scientists chit-chat online that there's a lot of concerns now about whether that science actually produced what the initial report was. Oh, did this engineering really happen? Is this a really robust result? So if I were covering that story, I would say, I got to keep an eye on that, because that may turn out not to be true. That was certainly a big splash. That's something that if it turns out not to be true, we should follow up on. So, following, whether it's Twitter or blogs, or there are various places where scientists talk about these things. There's something called PubPeer, which, if you're actually getting deep into this world, it's a really handy thing to know about. It's basically a place where scientists go and if they have a concern or a question or whatever about a particular paper, they'll publish a note on PubPeer, and they can do it anonymously, which is both helpful and dangerous, because they may be going after somebody they don't like, or they may be afraid to speak up in front of some powerful person in the world of science where they inhabit. But you have to be careful. You don't really know necessarily what those anonymous critiques actually mean. But PubPeer is a great place to go to sort of hear the chatter about a particular paper in the scientific literature. So just a tip on those points. So at any rate, these are problems that we can't solve as journalists, but these are problems that we can address, we can be aware of, and we can use them, and we can use a bunch of techniques to make sure that we're not overselling things, that scientists aren't overselling things, and that we're keeping in perspective that how science is actually built. It's piece by piece, small step by small step, and we need to find creative ways of telling dramatic stories about small steps. It's a challenge that we all face, but I think that's what we need to do. So thank you very much for your attention. Happy to take questions. And there are microphones here, and since this is being recorded, if you have a question, please come down to a microphone.
Hello, my name is Sahar Shmace, and I'm a second-year journalism student. I recently did a story about alternate methods to CRISPR, and I find science very, very fascinating, but I'm not ever going to be smart enough to be a scientist. So I was just wondering, how would a journalist approach doing scientific reporting when they find themselves to be kind of like not able to understand the actual scientific research?
Yeah, that's hard. And the first thing I would say is talk to as many people as you can and fact check as much as you can what you've written. One of the great science journalists of all time is a guy named David Perlman, who's 98, and he just retired this year from the San Francisco Chronicle. And I have crossed paths with him many times over the years. And one question he asked at press conferences that I always thought was tremendous was he would stand up and say, "Let me make sure I understand this." And he would put something into his own words and ask the scientist, "Am I getting this right?" Mm-hmm. And this is, I do this all the time when I'm doing phone interviews. What I liked about the fact that Dave was doing that was he was sort of the dean of science journalism in the Bay Area. And a lot of people who had less experience than in the audience could hear that. And it's like a question that anyone should ask. And people didn't necessarily ask because they felt like, I'm going to sound dumb if I ask that question. You have to get away from that idea. But if you end up with a story that you're not really sure about, I wouldn't press send on that story. I think if it's going to take a lot of work to get there, take the work, or at least start out with a simpler story that you have more confidence you can get right. I don't think it does anyone a service for you to put something out and cross your fingers and say, I hope I got that right. So yeah, but if you can find people who can help you explain these things. Over the years, I've developed a long list of people that I know in all sorts of fields, so I can call them up and say, I'm having trouble untangling this, who should I talk to, those kinds of questions. And those are just basic skills of journalism, no matter whether it's science or business or anything else, you need to do that. But yeah, it's good to know what you don't know also, and because that's really important. If you think you've got it all or close enough or whatever. That's sometimes where actually that's where kind of I make mistakes. It's like, oh, I know the answer to that, and I'm not double checking those things. And those are easy pitfalls even for very experienced reporters. So just that's a couple of ideas of how to proceed. But yeah, you chose the alternative CRISPR technologies as a particularly tough story for anyone to tell, and I give you kudos for taking that on.
Thank you. I actually got to speak with George Church, the person who actually conducted the alternate experiments.
Fabulous,
so,
yeah.
Yeah, I mean, he spoke with me in a way where he broke things down, but it was still very difficult after watching the lecture because of all of the terminology. So that's kind of what threw me off. And
it's wonderful to find a scientist like that who can really explain and break things down, because many scientists can't. And by the way, sometimes when scientists can't break things down, it's because they don't really understand it very thoroughly themselves. Honestly, scientists they're sort of comfortable in their little bubble, their little world of jargon. And if you say step out of your world of jargon, sometimes they just can't because they haven't thought more broadly about it. We need them to. So I tend to say thanks for your time, and I go find somebody like George Church who actually can do that with great skill. But it's sometimes it's a treasure hunt to find persons like that. So
thank you.
I have a question.
Please come to the microphone.
Okay.
Hi, I'm an old journalism professor, and now here's my question. What would your tips be for understanding a scientific journal article? You mentioned you should look at the p-value, but what are other tips? Because I think sometimes a lot of people don't go to the original source, they just rely on the press release from the university.
Right. It's a tricky one. I think that it really helps to have a scientific background to do this, because if you don't, you're going to get lost. Honestly, I think you're not going to be able to read a paper if you don't really understand the nuts and bolts of how science works. And I often get even lost in jargon, because every field has its own specific jargon, and you sometimes have to really struggle to understand. I may know the ideas in a paper and so on, but I'll come across a word. It's like, what does that mean? But my recommendation is certainly the best science journalists do try to read an entire paper. I may skip over the methods section a little bit, but sometimes at my peril. Sometimes I have a question that I know I want answered that's going to be in the methods section. I want to know how many people are involved. I want to know how good the statistics are. I want to know, the abstract sort of lays out what the results are, but the introduction often provides the context that I like to know in order to understand, why do I care about this? Where does it fit into other science? Because often in the introductory part of a scientific paper, they'll say, there's been this dispute over all these years, and now we have the answer finally, or we have this new advance that helps, that we argue supports this hypothesis over that hypothesis. And so that helps me frame my own story about what it's about. The other thing I do often when I'm looking at a scientific paper is, if I'm not sure exactly who all the right people are in the field, which I often don't, I will look in the references, because often in those introductory parts they'll refer to the big review papers or other big findings that have sort of led up to this moment. And then I look to see who wrote that, and it's like, oh, I'm going to call that person. And often they talk to the people who disagree, or they cite the people who disagree, and so I want to talk to them as well and say, okay, so now do you believe this, or now do you accept that this question has been answered? So those references, I look carefully at the footnotes, particularly of those cases, to find other people to talk to about those papers. So those are a few tips.
Okay. So are there any other questions? Well, thank you.
Yes, there's a question.
Hi, my name is Marino, and I'm a fourth-year journalism student. For NPR, you do like 3 to 4 minute or sometimes a little bit longer segments. So I was wondering how you take something so complex, sometimes research and stuff, and put it into those segments, and how you make sure that you're conveying new information in each session, and also making sure not to lose people's interest, because I'm always very in tune because you go from one thing to the next.
Yeah. It is the art of radio, and it's what I like, and what also drives me crazy is having 3 or 4 minutes to tell a story. But I consider it a challenge for me, just the way I want to find a scientist who can explain sort of big picture simply. It's like, I know that I need to do that for a story. I can't afford to get lost in the details and just dump a lot of details on people and hope they can figure it out. I need to have a really clear idea of what the story is. A radio story has a beginning, a middle, and an end. It actually is, even though it's 3 or 4 minutes, a good radio story is structured much more like a magazine story, because you know if people are going to start paying attention at the beginning, unlike a newspaper article, you can expect they'll listen to the end unless they click off the radio because they're disgusted with what they're hearing. But that's an advantage of a radio story, is you can have that narrative arc. And I think that's one way to hold people's attention. That's why at the beginning of an interview I'll say, why do you care about this? Why did you think about this topic or whatever? What got you interested in this? And then the scientists themselves start telling stories. And so the challenge is to embed the information in a story of discovery and exploration by the scientist. I try to avoid numbers as much as possible because people can't remember very many numbers. I have this sort of funny test that I use. I ask myself, how much of this will somebody remember in 3 days from now? And then I say, and how much will they remember the New York Times story 3 days from now? It's like, I bet we're about the same. So even though I have much less real estate to work with, I think that I have the potential, at least, to capture the emotion of the human voice and to do some storytelling and so on, to have something that has as much stickiness as a longer article in a newspaper would have.
Right. Thank you.
Here's a question.
Hi, I'm Larissa Herald, and I am actually not a journalism student. I'm an environmental engineering student. And so I'm kind of, I guess, asking almost the converse of some of the questions we've had before, like if I have an interest in journalism and writing, and so if I want to be able to communicate what I might find or work on in the future, like what could I do to kind of transfer those skills, almost the reverse direction?
Yeah, well, I think the first thing you need to do is think about the language you use, because I think scientists easily, or anyone in a technical field, gets lost in their own jargon. It's very handy, right? It's a shortcut. It's a really easy way to communicate with other people who speak the same language. But the first challenge is to step back from that and say, how would I explain this to somebody who's not in my field? And people who spend a lot of time just thinking about these problems from within that narrower context can sometimes find it very difficult to find that broader language. But I think that finding that language also helps you think more broadly about the context, because it's, and if you can find that language, then you can also explain why you care about it, why it matters to the world. I mean, who cares? I mean, that's a question that you should be able to answer if somebody says, who cares? Or you should actually, before you have somebody ask that question, you should give an answer why you should care. So that's one thing you can do. But I think that's the most important thing, really, is just think about what language you'd use to explain what you're doing, and recognizing that you're talking to somebody who's, presumably smart, but doesn't have the language, doesn't have the context that you have. And, yeah, that's the challenge.
Okay, thank you.
People can line up for the mics. You don't have to wait for someone. Okay. So.
I guess I'll do the conclusion from here. Thank you very much. This was fascinating. You explained science and science journalism in ways that were so clear that we could all understand it. So thank you very much. And thank you, AAAS and the Kavli Foundation Journalism Award lecture series for this. This has been awesome. Let's give them a hand.
Thanks for your attention.